Okay, so we're going to get started. Okay, so this is test 3, and yeah, so this is all on transcription. Okay, so I have the test, and the format is as follows. There are 39 multiple choice questions, and there are 7 free response with an extra credit question. So there's 3 points of extra credit, and it's all based on your participation in class. So if you've done any of the class, like his little models or whatever, or if you've asked questions in class, like things like that, will earn you up to 3 points. Okay. Like if you've ever asked a question, like you'll get a point for that.
How does he know?
Because he just hopes that you're honest. Yeah. So I mean, like if you haven't asked a question, then on Thursday ask a question, right? Or like since he's had a bunch of participation like things, if on Thursday you haven't participated before, you only need to like participate in something once. Maybe say, hey, I remember we did something with this. Can we do that again? It really helped my understanding, okay? And then people that haven't participated, get your butts up there because, you want those extra credit points. Okay. So these are the condensed slides. So I got the test like at 2 today, and so I've been working on the slides since then. So they just went up like 3 minutes before this review. So this is what we have. Thank you. But we're going to get started. Okay, so the first thing that I put on my slides is like opportunities for regulation. So when our cell encounters some sort of stress, it can respond to that stress with proteins. With enzymes, okay? And we have different places where we can regulate where we start to make up these proteins. So, for example, we can start all the way back with transcription, like maybe we encounter something and we say, okay, let's begin transcription to make mRNAs that are going to be translated into proteins to combat this certain type of stress. That takes a lot of time to do, but it's very energy efficient to only make the proteins when you absolutely need them. But maybe you want a very quick response, and so a quick response would be making the proteins all the time, which takes a lot of energy, but when you see the stress, bam, the proteins can act, those enzymes can act, okay? So these are the opportunities for regulation that we have. So transcription initiation, so we can control it. We do that with our transcription factors. RNA processing, RNA stability, protein synthesis. We can have our proteins already made. We can modify our proteins. Yeah. No. I think you won't have to reproduce it if that's what you're asking. But you will need to know that energy efficiency starts all the way back at transcription, okay, and maybe modifying transcription or modifying mRNAs. So that's the most energy efficient, okay? I think you'll be able to recognize it if you saw it in like a multiple choice question, right? Okay, because that's probably where you would see it. Okay, so you have transcription. He uses the term also DNA-dependent RNA synthesis, so don't get messed up. It's the exact same thing. Okay, in both eukaryotes and prokaryotes, so in yeast and in bacteria, synthesis requires a template. The new RNA is going to be synthesized 5' to 3', just like DNA. And just like DNA, it also needs a template. Initiation is going to happen from a specific DNA sequence. This sequence is called the promoter. You have a promoter in both prokaryotes and eukaryotes. We use an RNA polymerase. Obviously, it's different between the two. But the RNA polymerase, he reads 3' to 5'. Okay? And then in eukaryotes, the main RNA polymerase is going to be RNA polymerase II. That's going to transcribe most of our protein coding genes. Okay, and I didn't put the number in there just because I thought it was fun. Like, it's important to know that it's RNA polymerase II that does most of the transcribing of the protein coding region. Questions up to this point? Okay, great. Okay, so here's the general transcription events. Again, I didn't put up this for like fun. You need to know these orderings. So you won't have to reproduce it, but if you saw a multiple choice question, you would need to know the order well enough to put them in the correct sequence. Okay? So you have binding of our RNA polymerase to the promoter. It's a closed complex. The DNA hasn't been separated yet. And then you're going to have this bubble formation form, so the complex is going to open up. So you're separating the two DNA strands so that our mRNA can be made off of one of them. Okay? Questions about this? Okay. Yeah.
So on the last slide, so is the open complex present in all three of those in the bottom?
Yes. So you have binding, closed complex, open complex, transcription occurs. Yeah. Yeah. Okay, and we call that promoter clearance. Okay, and so if you memorize these boxes, you should be just fine for the test. Okay. Okay. And so now I've broken up my PowerPoint slides a little more than he did in class. We're going to talk about bacteria first, and then we're going to move to eukaryotes. Okay, we're not going to mix the two. Okay, so in bacteria, we have our RNA polymerase. It's comprised of the beta and beta prime subunits, the omega subunit, and the alpha subunit. It is the beta and the beta omega subunits that have the active site. This is where we're going to make our new mRNA, or whatever RNA we're making. The alpha subunit is what's binding to DNA. It's also what is interacting with transcription factors. And then the omega subunit is going to help with assembly. So you should know, especially beta subunits, and you should recognize these two also. But mostly beta subunits. Yeah.
Do we have to know the difference between beta and beta prime?
No. Yeah. So the active site is between the two. Yeah.
So the beta and the omega, they both do what?
Okay, so the beta and beta prime, it's the active site where mRNA is going to be made. Okay? And the omega helps assemble the beta and the beta prime together. Did you have a question?
Active sites between the two?
Yes, so right here, this is where our new mRNA is being made, okay? Okay. Correct. Yeah, no, mRNA. Okay, so we have to initiate transcription, so the making of these mRNAs. And so we do that with sigma factor, okay? And our cells have a whole bunch of different sigma factors. There's a specific sigma factor that controls all of the different genes in the cell for normal growth, Okay, and that sigma factor is going to look for a specific sequence at the -10 and -35 box. You might have another sigma factor that controls a bunch of genes under a certain stressful situation. So the sigma factors are specific to a specific sequence at the -10 and -35 box. Okay? So the -10 and the -35 regions, those are called elements, and our promoter is made up of elements. So if you hear the word promoter or element, remember, those are DNA sequences. And -10 just means that it's 10 nucleotides before transcription occurs, before transcription starts. Same with the -35. 35 nucleotides before our genes start, where we need transcription to start. So a sigma factor that binds to those two sites there, here we have sigma factor 70, binds to those two sites, allows the RNA polymerase to be put onto the DNA in the correct configuration so that transcription can occur. Okay? So that's prokaryotic initiation. Questions about that up to this point? Yeah.
Will we need to know specific sigma factors or sigma 70?
No. But you should know that the promoter is made up of -10, -35, and that those are elements. And elements make up promoters, and both elements and promoters are DNA sequences. Yeah.
So looking at the diagram there on the right, so in the -35 and -10 boxes are like downstream. So like, I don't know.
Upstream?
Yeah. So
Yeah.
the RNA polymerase is going to go 3' to 5', right? To read it.
Yes.
So how is the promoter like? I'm not seeing how like is the gene to the right, but to the right you're going towards
Okay.
3'.
I'll answer your question. Okay. Because this model is a little sketchy. It's not sketchy. It makes perfect sense. But remember, we have a template strand and we have a coding strand. And the coding strand is the non-template strand. So this 3' right here is pointing to this outside DNA right here, which is the coding strand. So the gene right here is going to be identical from 5' to 3'. But what it's actually reading is this strand that's 3' to 5'.
Okay, cool. Thanks.
Mm-hmm. Yes.
And you said that's RNA poly 2?
Yes. So this is our RNA polymerase 2. Well, no. So this is in bacteria. RNA polymerase 2 is in eukaryotes, but it doesn't matter. Okay. So bacteria, they don't have like all the different like junk DNA that we have. They're a little more simpler. You won't need to know anything about this, like the name of him or anything. Yeah.
So the sigma factor will bond at the -10 and the -35 site?
Correct. Okay. Okay, so then we start our synthesis. So our RNA polymerase starts to go. We have something called kinetic proofreading. So he's going to put in the nucleotides that are going to match the template strand of the DNA. And sometimes he might accidentally put in a wrong nucleotide. Now, if the RNA polymerase is going slow enough, then he can actually correct it right then. He can pull that mismatch directly out right away and then put in the correct thing. That's called kinetic proofreading because it's based on the speed. Okay, so he's moving along. He puts down the wrong thing. He's like, oops, that was wrong. He takes it out, puts down the correct thing. Okay, it's based on his timing. Okay, so that's kinetic proofreading. Then we have, yes.
Is that the one that has to do with like the pyrophosphate, or does that matter?
It doesn't matter for the test, but yes. Then we have the nucleolytic proofreading. And this one is where it makes a mistake and then it continues on. Okay? And then after it makes its mistake, it backtracks and it has to pull out all of the nucleotides that it's made and then start from where it made the mistake over again. Okay, so that's nucleolytic. So kinetic is based on speed, nucleolytic is not. Nucleolytic has this internal nuclease. Basically, I put the information on the slides that's most pertinent, whatever the word is. You know, thank you. I can't really, speak well. But yes, it's all there. Okay?
When you say
Yeah.
internal nucleus, you mean endonucleus, basically?
Yes.
Okay.
But we're just going to call him an internal nuclease. Yeah. I don't know why. That's how it was on his slide, so that's how we're going to call. That's what we're going to call him. Yeah.
With the kinetic proofreading, when it says it's dependent on the rate of reaction, what exactly does that mean? Does that just mean if it's going too fast, it'll just keep going and come back?
Yeah. So if it's going too fast, then you're going to have more nucleolytic proofreading. And to be honest, mRNA doesn't do that much proofreading to begin with, because if it makes a mistake, who really cares? The mRNA is going to be degraded soon anyway. Okay? It'll make more later on. Okay, questions up to this point? Okay, let's move on. Okay, so intrinsic termination, I'm just going to go ahead and draw this out on the board, because you're going to have to draw it out, possibly. So you have your DNA. Okay. And this is my 3' end. Here's my 5' end. Make sure it's that. Okay, and we have our 5', the 3'. Okay, so this is my coding strand. My RNA polymerase is going to use the template strand. Okay? And maybe my gene is going to start right here. So I'm going to draw my RNA polymerase like it's done a little bit of work. Okay? So this is my RNA polymerase. And he has synthesized quite a bit of mRNA. Okay? Okay. Okay. So in my template strand, we have these G's and these C's. Okay, it's a GC rich region, and it's just been transcribed. Okay, so it's right here. Okay, so we're gonna have C, G, C, G, G, C, C, G, C, G. Okay, so we get this GC rich region that's moving out of this RNA polymerase. Okay, over here in our template strand we have this string of A's. Okay, and our RNA polymerase is going to start putting in the mRNA strand a bunch of U's. Okay. Now, what do we know about U's and A's? How many hydrogen bonds are between the two? Two, right? So we get these 2 hydrogen bonds between each of them. And our RNA polymerase is going in this direction. So as this string of C's and G's comes out of the RNA polymerase, they're going to start to make a hairpin. Okay, so this is going to bind with that. Okay, now how many hydrogen bonds are between G and C? 3. So we have 3 hydrogen bonds. So what this ends up looking like is this hairpin right here. Okay, we get these hydrogen bonds between each of the nucleotides. Now, since we have these A's and U's over here, and these G's and C's that have just snapped together to form a hairpin, these G's and C's will snap so quickly together that the force will pull this string of U's out of the polymerase. Okay, do you all see how that happens? And that's called intrinsic termination. Okay, so in the DNA template strand, we had G's and C's and we had A's, so that our mRNA would have a GC-rich region to make a hairpin, and then a string of U's that could be pulled out of the RNA polymerase. Okay. Now, if we didn't have this region of G and C's, maybe it was just like A's and T's and G's and C's and whatever, then this hairpin that would be formed, it wouldn't be strong enough to break the hydrogen bonds between the A's and the U's. Okay? And in that case, this would continue to go out, and this guy would continue to transcribe more mRNA. Okay? It wouldn't terminate anything. Does intrinsic termination make sense now?
Yes.
Yeah.
So like on the test, he suggests like writing steps, like drawing steps out is kind of
Yeah, so maybe like start with what I had first, and then do like an arrow, and then show the same thing except with the hairpin, and then show like the force. Okay, if it was on the test, of course. You know. Yeah. And make sure that like if it was on the test and you were like labeling things, that you label like down here, this is my 5' end, this is my 3' end on the mRNA. You show your DNA primes. And I only say that because I don't know what he has in his answer key to have people like have points docked off their tests. So for that reason, I would just label everything you can. Okay, so that's intrinsic termination. We also have row-dependent termination. This is like very similar. But we don't have this string of A's and U's. So we get, this is my mRNA, but we get this site that's encoded into the mRNA, and it's called the RUT site. And an RNA helicase called Rho helicase is going to jump onto the RUT site. So he jumps on, and he starts moving up the mRNA. But the RNA polymerase, he's also continuing to transcribe mRNA. But eventually, he will make, in the mRNA, nucleotides that will be able to form a hairpin, a GC hairpin. So maybe I should put some G's and C's here. Okay. And this hairpin will be strong enough to slow the RNA polymerase down. Now, since there's no string of U's and A's here, it won't pull the RNA out of the polymerase, but the GC hairpin will slow it down enough for Rho helicase to catch up to the RNA polymerase and then hit him off the mRNA. Okay? In Rho helicase, he doesn't care about hairpins because he's a helicase. He can unwind them himself. Okay?
Say that again, sorry.
Rho helicase, he doesn't care about hairpins, like it's not going to inhibit him. When he sees that hairpin, he just runs over it. It doesn't matter to him. Okay? So he can catch up to the RNA polymerase and push him off the mRNA. Okay? And this is also a form of termination. So this is row-dependent termination. Okay? Yeah.
Is this one we have to draw as well?
Probably not. Yeah. Okay. So now we're going to talk about the lac operon, which you should know pretty darn well. Okay, so we have our bacteria cell, and what is it? What? In Portugal.
Oh, in Portugal.
Okay, so we have our, yes.
Are you still giving a review on Saturday too?
Yes.
What time is that?
I don't know. But I'll have that posted by tomorrow. If not, come and complain to me in class, and then I'll post it. So here's my bacteria cell. And my bacteria cell, he eats glucose. Okay, so Glucose is represented by the stars. Now, here's my piece of DNA. Remember that we have like those genes called housekeeping genes? They're always making specific enzymes. Some of these enzymes that they make all the time are enzymes that are used to break down glucose. Okay, so enzymes of glycolysis, enzymes of the citric acid cycle, it's always making these enzymes. Okay, so bacteria, they prefer to eat glucose. Okay, now, also on this DNA, we have this region of genes that encode proteins or enzymes that will break down lactose. So maybe I have lactose present. So that's lactose. Now, both glucose and lactose can be broken down for energy, to make ATP. But if we are already making the enzymes for glucose, if we have both glucose and lactose, we're going to prefer to use glucose as our energy source, right? Why take energy to make up the enzymes for lactose when the glucose enzymes are already there? Correct? Okay. But if glucose all of a sudden disappears and we only have lactose, now it becomes essential to make these enzymes that are going to break down lactose. Now, when we break down something, it's called a catabolic process. So the breaking down of lactose is catabolism, right? So I have that right here. Are we good up to this point? So we need a system in our bacteria cell that says, okay, is glucose absent and is lactose present? Because likewise, maybe glucose is not present and maybe lactose isn't either. There's no reason to make a lactose enzyme if lactose is not present. Okay? So in my slides, I've broken it down into the two sections. Okay, so the first one is detection of the lactose levels. So, here is my DNA. Okay, here is my promoter. So remember, to my promoter, we're going to have sigma factor in the RNA polymerase bind. Okay, and around my promoter, I have something called an operator. Now, once again, an operator is a DNA sequence. And I can have a repressor that binds to my operator. So in the case of the lac operon, okay, so these are all the genes for lactose metabolism enzymes. So in our bacteria, the operator is going to be bound by a repressor. Okay? So here's my repressor. Now, the repressor will only fall off if he binds to a sugar called allolactose. Okay? And allolactose is a derivative of lactose. Okay? So, if lactose is present, then allolactose is high. Okay? And if allolactose is high, it'll bind to the repressor. So, my little allolactose molecules right there. And the binding to the repressor will cause it to fall off. Okay? So my repressor now falls off. So the repressor is negative control that is determining if lactose is present or not. Okay? So repressor goes with lactose levels in the lac operon. Questions so far up to this point? Okay. Now, just because the repressor is gone, we also need an activator to bind so that we get transcription. And so we have a region on our DNA. It's a DNA sequence also. It's called the enhancer. And what happens is we have this activator that's called CRP. Okay? Maybe I should. Oh. Here is by the slide. This is the slide of everything I just said. There's my repressor. He's fallen off because allolactose has bound to him. Okay? There he is. Okay. And so now we're going to look at our activator. Okay, so we have CRP, and we also have, so we're looking at glucose levels. So again, if we have glucose present, there's no reason to make these genes that are going to be made into enzymes to break down lactose, because we'll just break down glucose instead. Yeah?
So if glucose and lactose are present, wouldn't allolactose also be present if you take off the
Repressor. But we still wouldn't have transcription because we don't have the binding of the activator. Okay, so in this case, in the lac operator, you have to have the repressor fall off and the activator bind. And so when glucose levels are low or not present, that means that we have high levels of cyclic AMP. Okay? And when we have high levels of cyclic AMP, then cyclic AMP can bind to our CRP, and then the CRP will bind to the enhancer, and now we will get transcription of all of these genes, which will make mRNAs, which then will go through a ribosome, which will be made into enzymes. Okay? So one part is looking at glucose levels, another part is looking at lactose levels. Now both our repressor and our lactose, both our repressor and activator, they required the binding of a molecule to either fall off or to bind. So CRP could only bind when CAMP is bound to it. Okay, so that makes CAMP an effector molecule. Okay, so that's the term for him. Whereas in the case of the repressor, when allolactose binds to the repressor, he falls off. Okay, so allolactose is also an effector molecule. Okay, so the binding of it changes whatever it's doing. Yeah.
Does that make CRP an apoactivator?
Yes. Yeah. But you won't be asked that. Yeah.
So to have expression, we need to have like lack of the negative control and the positive control.
Right. Yes. So in this case, it's positive control that's looking at our glucose levels. Yeah.
So, okay, so that is positive control because now you're working with 8 different systems.
Yes. I mean, it's all controlling the lac operon, but this is independent of what's happening with the repressor. This is based on glucose levels. The repressor is based on lactose levels. Yeah.
So we want high lactose levels and low glucose levels for this to occur?
Correct. Yes.
So when the repressor is on there, and the process of removing the repressor off of the allolactose, that is negative control.
Yes.
Regulation, whatever. And then this is positive regulation because this CRP is going to cause something to happen.
Yes. That's correct. When you guys are thinking about like positive and negative control for this class, I think the easiest way for the test is to say, if the certain protein is bound to the DNA, what is its effect? If it's causing it to go, it's positive. If it's bound and it's causing it to stop, then it's negative. Okay? So this is a positive control system right here. Okay, when CRP is bound, we get transcription. Yes?
So what would be the difference between an effector and a cofactor?
Okay, so when you're talking about a co-transcription factor,
Yeah.
in that case, the co-transcription factor is actually the one that's binding to the RNA polymerase.
Okay.
Okay, so the activator is going to bind to the DNA, and a co-activator to bind to the activator, and then bind to the alpha subunit of the RNA polymerase. Other questions? So, yes.
Just a little bit confused about the negative and positive control because on here it says that when L-lactose binds to the repressor, it's negative. I know that still needs the operator, or the operator to be going in order for it to actually happen, but how is the L-lactose binding to the repressor negative?
So it's looking at the whole system. What does the repressor do? The repressor, he causes transcription to stop, and that's why it's negative control. So just because the repressor falls off, now it has the opportunity for transcription to start, but it's not going to start without something activating it, causing it to start. So the only thing the repressor can do is stop transcription. He can't do anything else, and that's why it's negative control. Yeah.
So kind of go along with that, sometimes in class you said that even if you're having transcription occur, it can still be a negative process.
Yes.
And how's that? It's just because it's being shut off or like slowed down.
Because whenever this binds, then we don't get transcription. If this molecule is bound to the DNA, transcription will not occur. So this molecule causes negative control. If it binds, no transcription. Okay? So it's negative control, even if it's not bound, then of course we're going to get transcription that occurs. But once it binds, there's no transcription. Okay? Because it's a negative controller. Yeah,
I thought allolactose knocked off the repressor.
that's correct. Yes.
Can you point out like the enhancer on this?
So in this one, we don't have an enhancer, we just have the operator. But in this guy right here, where you see CM binding, that's my enhancer. Yeah.
What would you define an effector as?
Any molecule that binds to a transcription factor and changes its binding capabilities to DNA. So it either makes it better or it makes it worse. Yes.
Would you say that repressors only bind to operators and activators only bind to enhancers? Is that accurate?
Yes. Yes.
A second ago you said transcription won't happen if allolactose, it's negative. I thought allolactose, it binds to the repressor, knocks it off, so will transcription happen?
That's correct. But if the repressor is bound, if there's no allolactose and the repressor is bound, then we don't get transcription. Okay? So it's still negative. Yeah.
The distinction basically just comes down to whether the allolactose is bound or not. Like that repressor is not going to cause transcription to happen. It's either going to allow it or not going to allow it. Whereas with the positive regulation, that activator is actually going to cause it to happen.
Correct.
Is that the distinction?
Yeah. Another good distinction is if it's called a repressor, then it's negative control. If it's called an activator, it's positive control. Okay, we're going to move on from the lac operon, and we're going to go to the trip operon. Okay? So, tryptophan is an amino acid that our cell needs for protein synthesis. So tryptophan will be incorporated into growing peptide chains. Okay, and sometimes our cell it lacks tryptophan, and so it can actually build tryptophan up. But in order to make the tryptophan, we have to make the enzymes to make the tryptophan. Okay, So since we're building up tryptophan, that's the end goal. This is an anabolic process, okay? Okay. So this one's quite a bit easier, I think. Because we're just looking to see if tryptophan is not present, then we need to make these proteins, these enzymes. So, we have this repressor, okay? Here's where my tryptophan genes are going to start. So these are all the enzymes that are going to make up tryptophan. Here's my repressor right here. And he doesn't bind to my DNA, okay, alone. So he's just kind of sitting here. So if there's no tryptophan, we're going to get transcription, okay? But now if tryptophan is present, we don't want transcription because we don't need to make the enzymes to build up tryptophan. So what happens is tryptophan will actually bind to my repressor, okay? And the binding of tryptophan makes the repressor bind to my DNA, which turns off transcription. So tryptophan is present, repressor binds. So that's pretty simple. This guy's called, if you see the word in your test like aporepressor, this guy is an aporepressor, okay? Because the binding of the effector made the guy bind to the DNA. So if you're increasing the ability to bind to DNA from adding an effector molecule, then it makes an apotranscription factor, okay? So if it makes an activator bind better, it's an apoactivator. If it makes a repressor bind better, it's an aporepressor.
So the repressor itself is an aporepressor, or the tryptophan molecules?
No, the repressor itself.
So why does it bind better?
Because when tryptophan binds to it, it causes the repressor to change shape just a tiny bit so that it can now bind to the DNA. Okay?
Which one's the effector?
The tryptophan. Yeah,
So CRP and apoactivator.
correct. But he won't use the term on the test. Okay, so now, since we're talking about a repressor, and when it binds, we don't get transcription, this is negative control. Okay? So that's one way that we can control transcription of the lac operon. And the other one is called attenuation. Okay. So I'm going to draw this guy up here. So here's my DNA. And we have my RNA polymerase. And he has made a bunch, well, he hasn't made a ton. He's making the leader sequence right now. Okay? So my genes that are going to make all those enzymes to build up tryptophan, he hasn't even gotten to those genes yet. Right now, he's just making the leader sequence. Okay? So what happens is this mRNA that's growing, this leader sequence right here, it has these two codons for tryptophan. Now, something that's different between bacteria and eukaryotes is that bacteria, they don't have a nucleus, okay? So once this mRNA is being made, a ribosome is going to hop right onto it and start making a protein, okay? So here is my ribosome, okay? And he starts making this junk protein, okay? So he's putting in these amino acids, okay? And he gets to this tryptophan codon that says, okay, ribosome, the next amino acid to put into this growing peptide chain is tryptophan, okay? Now, if tryptophan is present, he puts it in very quickly. And he moves on to the second codon for tryptophan. And since tryptophan is present, he puts that tryptophan in. Okay? Now, part of the leader sequence are these 4 regions right here. Okay? So we got region 1, 2, 3, and 4. Okay? Now, if he was able to put the 2 tryptophans in quickly, so here, these are right here, the RNA will, or the ribosome will actually move over regions 1 and 2 very quickly. Okay, so now he's right here. This is my ribosome. Okay, so he moved over those 2 regions very quickly. And now 1 and 2 will make a hairpin, and 3 and 4 will make a hairpin. Okay? Now, the 3-4 hairpin is GC rich. Okay? Now, part of this leader sequence, and this looks very much like attenuation, is the synthesis of a string of U's. Okay? So if this 3-4 region makes a hairpin, it's going to snap together very quickly, and it's going to pull this mRNA out of the RNA polymerase, terminating transcription. Okay? And then we don't get the transcription of all of these genes further downstream. Does that make sense? Now, if the ribosome gets to these 2 trip codons and tryptophan is not present, then he stalls and he sits there until he gets maybe the 1 tryptophan molecule, there's more than 1 tryptophan molecule, but tryptophan is low. So he sits there for a while looking for a tryptophan. Okay? And what happens is that allows for the 2 and the 3 regions to move together and form a hairpin. And 2 and 3 is not GC rich. Okay? And since 2 and 3 are not GC rich, it won't pull this string of U's out of the RNA polymerase. Okay? So, the 2 and 3 hairpin, that's called an anti-terminator. And the 3 and 4 hairpin is called the terminator. Yes?
Will he ask us to label them as like positive and negative?
He won't ask you to draw this. But you need to understand it well enough that, let's say that he says you delete the tryptophan codons in the leader sequence. What's that effect? Well, if you have no tryptophan codons, this ribosome is going to move very quickly. What's going to form? Which hairpin? 3 and 4 would form, right? Because it would move right over the 1 and 2 very quickly. And that would mean that you have a terminator, and you wouldn't get transcription of all of these genes to build up tryptophan. Or maybe if you delete these A's right here so that you no longer get a string of U's, what would be the effect? Well, if you delete them, this guy's not going to get pulled out. If he doesn't get pulled out, the RNA polymerase will continue on, no matter what hairpin is formed. Okay, so you need to know this well enough so that if he changes one little aspect of it, you can answer questions about that. Yeah.
When 2 and 3 form a hairpin, would 1 and 4 also combine together? Or
No. Yeah.
When 3 and 4 form a hairpin, would 1 and 2 also form one?
1 and 2,
It just
3,
doesn't matter.
3 and 4, yes. So if 3 and 4 form a hairpin, 1 and 2 are forming a hairpin too, because the ribosome's going to sit in the middle of them.
But
Yeah.
that 1, 2 hairpin doesn't really do anything.
No, it's the 3 and 4 that's the anti-terminator. Yeah.
If there are tryptophan codons, transcription happens, right?
The codons are present. If there is tryptophan that's incorporated, then transcription will not occur.
Because with tryptophan, the repressor falls off because 1 is 2
Correct. Yep. Yeah.
During this whole process, if tryptophan is not present, the ribosome is just chilling on the leader.
Yeah.
So it's not like translating or anything.
Okay, so your cell will never run completely out of tryptophan. It's just that we say that there's no tryptophan. In reality, it's just in a very low concentration. So eventually he'll grab a tryptophan. He'll go to the next tryptophan codon. He waits there. Finally, he grabs one. But 2 and 3 have already formed a hairpin, which is an anti-terminator. And now he can move on and make the rest of the enzymes that are going to build tryptophan.
Okay. It's polymerase that's getting the RNA ripped out of it, right?
Yes. Correct.
So it's the same as intrinsic termination.
This is very similar to intrinsic termination. That's correct. Yeah.
So basically, just in this whole thing is, if the codons for tryptophan are there and led by the ribosome, you get ineffective, or you get terminated 3 to 4 hairpin. If they're not there, it lets it kind of stall and go, and you get the 2 to 3, which is angus.
Correct.
And that allows transcription, right?
Right. Okay. Yeah.
Sorry, I know you already mentioned this, but like let's say there was tryptophan, but there's no U strand.
Okay.
So like there's hairpins to stop the ribosome,
But there's no use, so it won't pull out. So the RNA polymerase would continue on.
I can go through this hairpin.
Well, the RNA polymerase, it doesn't need to.
Actually, yeah, my bad.
Yeah, the ribosome can go through the hairpin.
Yeah,
It's just fine. Yeah. Okay. So we're gonna move on. Okay. So riboswitches. You don't need to know a lot of detail about these. This is when something binds to RNA that causes termination. Yeah, that's a riboswitch. So the only one we talked about was the tyrosine riboswitch. And it works in a very similar way. And he really won't go into a lot of detail with these. So you just need to know the very basics. But what happens is, okay, I don't know why I just did that. In the tyrosine riboswitch, these genes are for making enzymes that are going to build up tyrosine, okay? And tyrosine is an amino acid. And amino acids bind to tRNAs, okay? tRNAs bring them to the ribosome so that they can be incorporated into proteins. So if tyrosine is present, we get this tyrosine that's bound to this tRNA. And this tRNA, he will cause an anti-terminator to form, okay? Anti-terminator will pull this guy out of the RNA polymerase so that we don't get synthesis of these genes that are going to make enzymes to build up tyrosine, which makes sense. Tyrosine is binding. We don't need more tyrosine, okay? Now, if tyrosine is in low concentrations, we're going to get tRNAs without tyrosine on them. And in that case, it's going to make a different hairpin. And this hairpin is an anti-terminator. It's not strong enough to pull the string of U's out of the RNA polymerase. This is still the riboswitch, okay? Yeah.
So just to clarify, when the tyrosine is there, then it forms a terminator loop, right?
Yes.
Okay.
Right. And the big takeaway here is, can you define a riboswitch correctly? Okay? Like, do you understand what it is? This is the only example of a riboswitch he talked about. Okay, but it could be any molecule. Maybe we're not talking about tyrosine, we're talking about some other molecule that binds and causes an anti-terminator. These are riboswitches. Yeah. The whole process overall is the riboswitch. Okay, so if you have the tRNA without the amino acid, and then you put the amino acid on, an anti-terminator is going to form. If you don't put the amino acid on the tRNA, anti-terminator doesn't form. But the tRNA with the amino acid, or just the tRNA itself, that is the molecule that we're referring to that binds to the RNA. Yeah.
It seems to me that this is really similar to the tryptophan, but it's doing the same thing, just like just a little p.
Yes, in a different way. Yeah. So in the tryptophan, we use a ribosome to control the bindings of the 3 and 4 regions.
So here we're using tRNA.
To make, yes, and the region is not the same in this. You don't have like the 3 and 4, 1 and 2. It's just a tad bit different. So in this, it's called a riboswitch. When we talk about the tryptophan, we're talking about attenuation. Yeah.
So tRNA, that whole figure, they're just the boot?
This is the tRNA right here. This is RNA, okay? And really, you just need to know the very basics of this. So don't like go studying it crazy. Just know what a riboswitch is, okay? This is pretty simple. You put a tyrosine on, forms the anti-terminator. No tyrosine, you get. Did I say form? Yeah. Put a tyrosine on, forms the terminator. No tyrosine, forms an anti-terminator. Okay. Good. Okay. Any other questions? Bam, there's my anti-terminator. Okay, so then lambda phage. Let me explain lambda phage a little too, because there is going to be some questions on that. So we have our bacteria. So we're still talking about bacteria. We haven't even gone to eukaryotes yet. Okay? But we're almost done with bacteria. So we get these like phages. It's my phage. Okay. And our phage, so here's my bacterial DNA. In reality, it's in a circle. Okay? And my phage, in this case, this is my lambda phage. He's going to insert his genome, okay, into the cell. In the cell, this bacteria cell has two options. One, it can take this genome and replicate it a bazillion times and then make a ton of phage from it, okay? So we make a bunch of phage. You get the idea, right? And then those phage, because you're making so many of them, they're going to burst the cell, okay? They're going to lyse it, okay? So if that happens, that's called a lytic lambda phage, okay? The other option that the cell has is it can take this inserted gene. It's not going to replicate it, but it can actually nick its own genome, and it can insert it into the genome, okay? And that's called a lysogenic lambda phage. So one is a lytic cycle and one is lysogenic, okay? So the question now becomes, how do we determine if it's going to be lytic or lysogenic, okay? And it depends on transcription factors, okay? And the ones we talk about are the lambda repressor and the CRO protein. Okay? So the CRO protein, he inhibits the synthesis of all of the genes that are going to incorporate this piece of DNA into the host. So CRO protein inhibits this so that it can't be incorporated. So then we're going to revert to being lytic. Does that make sense? Now, maybe instead of high concentrations of CRO protein, we have high concentrations of the lambda repressor. In that case, the lambda repressor will repress all of the genes that keep this piece of DNA out of the genome, okay? In that case, it will be incorporated into the bacteria's genome, and that'll make it lysogenic. Right, so if CRO is low and lambda repressor is high, okay? Questions up to this point of what I've said? Okay, now, when it's lysogenic, now the question is, how can we make more bacteriophage if it's lysogenic, right? We're not making any. And so our cell, it has what's called an SOS response, okay? I shouldn't have drawn that there. Okay, so we get these genes in the actual bacterial genome. And these genes, they will be made into mRNAs, which will be made into proteins that will tell the cell to kill itself. Okay? So they promote apoptosis, which is a very organized, clean way of killing itself because it's saying, I'm damaged, I shouldn't multiply. Okay? And so we have all these genes in this bacteria here, but they're not being expressed because we don't want the bacteria to just die all the time. And so we have a repressor on these genes. And that repressor is called LEX-A. Okay, so they're all controlled, all of these genes are controlled by LEX-A. Now, do you remember what it's called when you have one transcription factor controlling a bunch of genes?
Global control.
Global control, right? So this LEX-A system is global control. Okay? Now, when we get the SOS signal, so there's damage to our DNA, we need this cell to die. We don't want it to make offspring. What happens is we have, so you remember from the last test we had RecBCD, which recognizes DNA damage, okay? And you have like RecA. So these will help break up LEX-A, okay? So LEX-A breaks up. Okay, maybe this is a little too long of an explanation. Don't worry about memorizing RecBCD. You won't see it again. But the big takeaway is that the lambda repressor, okay, which is keeping this in the lysogenic phase, okay, so you have some genes on here, they keep it in the lysogenic phase, you have this lambda repressor bound to it. It also, if you have this SOS response, it, like the LEX-A, will be broken up, and now the repressor, it doesn't work. And if the repressor that's keeping this in lysogeny doesn't work, then we become lytic. Yeah.
It has an SOS response to stop it from reproducing. But when you have the SOS response, it then allows it to be reproduced.
No, okay, so I'm sorry. I wasn't clear. So SOS is to stop the cell from reproducing. Okay. Now, it breaks up LEX-A so the cell can't reproduce. In the process, it breaks up the lambda repressor. So now the bacteria, not the bacteria, but the phage can reproduce. Because if the cell is going to die, the phage is saying, I need to get out of here. Okay? So the phage now is going to reproduce a bazillion times, and it's going to jump ship. Okay? Does that make more sense? Okay. These are the big takeaways. You have the lambda repressor that inhibits genes that would make the phage lytic, so it becomes lysogenic. So if you break up the lambda repressor, it's not going to stay lysogenic. Okay? And you break up the lambda repressor when you have an SOS response. If you have the CRO protein in high concentrations, it's going to be lytic. Okay? Okay, so that's lambda phage. Okay, so we're done with bacteria. Okay, now we're moving on to eukaryotes. Yeah.
Wait, lytic is when it's going to die, or lytic's when the cell's going to die?
What is it?
Is lytic when the cell's going to die?
Yeah, the cell will die if it's lytic. Okay. So, like bacteria, eukaryotes need a promoter in order to start transcription. Okay? So, these are the elements that make up my promoter. So remember, elements are DNA sequences, promoters are DNA sequences. These are all DNA sequences. Okay? So we have DPE, we have initiator, we have the title box, we have the BRE. Okay? Those are all elements that make up our promoter. And so what happens is this guy is like our sigma factor. He's going to bring the polymerase in. Now this is RNA polymerase 2 now, okay, the one that does most of the protein coding. So we have transcription factor 2D, and he's going to help align our RNA polymerase. Okay, we have other helpers. So we have the TATA binding protein, which binds to the TATA box. We have BRE, which binds to our transcription factor 2B. Okay, those can also help align RNA polymerase. Okay, so it's important that you know this figure. Okay, that you know what the 4 elements are: BRE, TATA, initiator, and DPE. Okay, and notice that initiator is where the actual gene is starting. Okay, so the important protein coding region starts right here. Okay. Yeah.
Do we need to be able to draw this?
No, but well, you need to know it very well. So I can't remember if it was like a multiple choice thing or if you draw it, but I think you really need to know it well, or I wouldn't have put like a whole picture to it. Yeah.
So the TATA box, BRE, the main function that we need to know is that they help align the polymerase.
Yeah, and you need to know, you guys need to know TF2D, TF2B, and TBP. Like you need to know those proteins and their names well enough to write them out. Yeah.
And so basically, TF2B is going to bind to the BRE sequence, and TBP is going to bind to the TATA box sequence.
So TBP binds to TATA. Is that what you just said?
I think so.
Yeah, because TBP is TATA binding protein. That's what it stands for. And then transcription factor 2B is going to bind to the BRE and the TATA binding protein.
So TF2B binds to both of them.
Yeah.
Do we need to know anything about these numbers?
No. But you should know, like, you should know that the initiator is where the gene starts, that DPE is into the coding region, that these guys are before it. Like, you should know those characteristics. Yeah.
I don't imagine we need to know like the numbers.
No.
But what if
Probably
the
not.
slashes important?
That's just representing the gene.
Okay.
Did you say 2B binds to both of them?
TF2B is going to bind to the BRE and to the TATA binding protein, okay, which is bound to the TATA box. Okay? Yeah. Which one? I'm sorry. Oh, DPE. Yeah. So this is an element that TF2D can bind to.
TF2D helps align.
Correct. Yeah.
TF2D is also bound to TF2B, right? So in all, TF2B is bound to 3 things.
Yeah. That's correct. But I don't think you need to have this. Like he said in class that you only need to have 2 elements for transcription to occur. But you should just know this whole picture though. Yeah. Okay. Yeah.
So those are all each separate elements, but they're also DNA sequences.
Yeah. Elements are DNA sequences. And these elements make up the promoter. Yeah.
So when you said know the name, like TF2B, just that or the actual?
No, just TF2B. Yeah. Okay. Okay, so here's some of the proteins of eukaryotic transcription. I've just put them here because you'll probably just see like definitions for them scattered throughout the test. So here they are. You have activators, they activate. You have repressors, they repress transcription. You have co-activators, which are proteins that bridge between activators and the RNA polymerase. You have co-repressors, so they're proteins that bridge between repressors and the RNA polymerase. You have mediator, which is a type of co-activator. it takes all of the different activators, and it makes it so that they can interact with our RNA polymerase. So that's mediator. So he integrates all the signals from all the different transcription factors. Because remember that in eukaryotes we have combinatorial control, where you can have multiple transcription factors controlling a gene. So it's no longer just like one activator and one repressor. You can have 3 or 4 or 5 or 6 transcription factors, activators or repressors controlling a gene. Okay. You have insulator, makes it so that transcription factors that are supposed to be affecting one gene don't affect another gene. Okay. So these are just kind of the basic definitions of some of the proteins in eukaryotic transcription. Yeah.
I thought the insulator was DNA. What is it in class?
You might be right. You're probably right. It's probably the insulator binding protein. So write it down and ask him in class tomorrow just to double check. Yeah.
Is that red, a little red, I guess we call protein in the upper image? Is that a mediator? Or
No. So this guy is the mediator right here.
Oh, okay.
And I only put this picture in there, not because you need to know the picture, but because I just wanted to show you mediator. Okay? So he's just this big protein that can take all of these different transcription factors and help turn on this RNA polymerase. He integrates all the signals from repressors and activators.
What do you call like the DNA sequence that codes for like an activator to come in? An activator is a protein itself.
Right, so it's an enhancer.
What is a co-repressor again?
It's a molecule, so it's a protein that bridges the gap between repressors and the RNA polymerase. So it's the same thing as a coactivator, except for it bridges between repressors. Yeah.
So is the mediator basically like this big convenience store kind of guy that goes along and is like, oh hey, you need this? Well, I got it. I can help you, and then gives a random protein to whoever needs it. Like that's just a bunch of random stuff, and then can just divvy it out when it's needed.
Yes, to some extent. He will control the signals from all of the transcription factors that are between two insulators. So either in front or behind. Because remember that enhancers in eukaryotes are they're location independent. Okay? They don't care where they are because mediator will help them come closer to the RNA polymerase. Yeah.
I think this was answered earlier, but what would be the difference between just effector molecules and coactivators and co-repressors?
Just effector molecules, coactivators, and Okay, so when it's an effector, this is my activator. Okay, here's my RNA polymerase. The effector will bind to it, okay, and that causes the activator to bind to the DNA, and it binds to the RNA polymerase. That's the effector molecule. When we're looking at coactivators, here's my RNA, here's my activator, and my coactivator binds to the activator, and he's the one that binds to the RNA polymerase.
So coactivators and co-repressors will bond to both the activator and the RNA polymerase, whereas if it's just an effector molecule, it just bonds to that.
Mm-hmm. Okay. We also have combinatorial heterodimerization, which is basically it's saying that we can have two parts of our activator or a repressor. So here is my DNA. And we have like all of these subunits. So maybe this is A, this is B, this is C, this is D. And here maybe this is my enhancer region. So we can just make combinations of these. For example, we could put A and B together. They dimerize. And maybe they become a repressor. Or you can put A and C together, and maybe it's a stronger repressor, or maybe it's a weaker repressor. So we can do this combination of these two subunits that are going to dimerize together to make different strengths in activators or repressors. Okay? That's combinatorial heterodimerization. Yeah.
And you say that's going to be activators or repressors?
Correct. Yeah. Double check in class tomorrow, but I'm pretty sure it can be either one. Yeah.
What's the difference between the enhancer and the operator again?
Operators are where repressors bind, and enhancers are where activators bind. Yeah.
So in this case, you have repressors binding to the enhancer.
I'm sorry. I wrote enhancer. I didn't really mean that. Yeah. Thank you. Okay. So now that comes to the Gao gene regulation. So I broke this down just like our lac operon in bacteria, but we're talking about eukaryotic cells now. So I have here my yeast cell. Okay? And one big difference between a bacteria and a eukaryotic cell is the size, and the fact that the eukaryotic cell has a nucleus. Okay? So in this we have my DNA. Okay? And we make all of the proteins again in this yeast to break down glucose. Okay? So if glucose is present, we're going to use it for energy. Okay? But another sugar is called galactose. Okay? And even if galactose is present, if glucose is present, we still are going to break down the glucose. Okay? Because there's this series of genes right here which will make all of the enzymes that break down our galactose. Okay? So we only want those enzymes to be expressed when glucose is low and galactose is high. So we have this activator that is bound to our DNA. Okay, and this activator is called GAL-ADP. Okay, but to this activator we have something called a co-repressor. Okay, and this one is called GAL-4P. Okay, so GAL-4P is bound to GAL-ADP. So we don't get transcription. Okay, because of 4P being bound.
The co-repressor is the GAL-4P or the GAL-ADP?
Oh, wow. I wrote it wrong on this. I'm going to correct it tonight, so don't download it right now. Even though you all have it downloaded right now. Or you can change it yourself. Yeah. So it's the GAL-ADP that does the active. Am I right? Oh, yeah. Thank you. I'm wrong. It's GAL-4P. Sorry. And then the GAL-ADP is the repressor. Okay? So GAL-4P is the activator, GAL-ADP is the co-repressor. Okay? So this co-repressor is always bound to GAL-4P, but when galactose is present, so here's my galactose, there's a series of signals that tell GAL-3P to bind to ADP and 4P. Okay? And when 3P binds, it's a coactivator, GAL-ADP will fall off. Okay? And now that GAL-ADP has fallen off, GAL-4P can do its business, and we can get transcription. Okay? If galactose is present. Okay? So GAL-4P, ADP, 3P, those are all controlled by galactose levels. Are we clear up to that point? Yeah. What is it? Yes. So this is positive control. And the reason it's positive control is because you're looking at what's bound to the DNA, which is Gal4P, and what is this action when it's not inhibited. Okay? So although you have this co-repressor, ADP, the co-repressor is repressing the ability of Gal4P to do its job. Okay? Yeah.
This might be super confusing, but so is Gal ADP considered negative control and the whole system is positive control?
Yeah, don't go too crazy about thinking about it. Just know that this system is positive control.
So
Yeah.
Gal4P will initiate transcription, but ADP reverses it. When lactose is there, it brings in 3P, which shuts it down.
Exactly. So it brings it in, shoves it off, and now we have activation because our activator's there. Okay? Yeah.
Just to make sure your slide is correct then.
Yes.
Okay.
Okay. So that's the galactose levels. But then again, remember that if glucose is present, we already have the enzymes to break down glucose. There's no reason to build up enzymes to break down galactose, okay, or to import it into the cell. And so what happens is we need something sensing the glucose levels. In that case, we're going to have a repressor that does that job. So the positive control is controlling the galactose sensing. The negative control is going to control the glucose sensing. So let's get rid of this guy. So here we have our DNA again. And in our nucleus, we have this protein that's called TUP1. Okay? And now here we have something called MIG1. Okay? Now, when MIG1 gets phosphorylated, then MIG1 cannot enter into the nucleus. Okay? But if MIG1 is not phosphorylated, he enters into the nucleus, binds with MIG1 with TUP1, and those bind to the DNA as a repressor. Okay, so they dimerize together, and they bind and they repress the gene. Yeah.
So phosphorylation
Cannot enter. Okay? So the thing.
Unless this phosphorylation you enter.
Not be phosphorylated, so.
I think we're
When glucose is present, MIG1 cannot be phosphorylated. No, but it's supposed to say so it enters the nucleus. Yeah, so I'll take the can't out. Okay? Okay, so don't look at the slide, look at me. Okay? Yeah. So, if MIG1 is phosphorylated, he can't go into the nucleus. Now, if glucose is present, okay, so glucose is present, glucose will block MIG1 from being phosphorylated.
Dephosphorylated, right?
He will dephosphorylate MIG1, block it from being phosphorylated. So MIG1 is not phosphorylated. MIG1, no phosphorylation. So MIG1 enters the nucleus. Okay? Yes?
Did you say that's when it's high glucose?
When it's high glucose. And this makes sense because if it's high glucose, we want to repress the genes that are going to make enzymes to break down galactose because we're going to break down glucose instead. So high glucose, no phosphorylation of MIG1. Okay? Now, if glucose is not present, it cannot block the phosphorylation of MIG1. Okay? So the phosphate group will bind to the MIG1, and now it can't go into the nucleus. And if it can't go to the nucleus, it can't dimerize with TUP. And if it can't dimerize with TUP, now you don't get any repression. Okay? So if there's no glucose, MIG does not enter the nucleus. Okay? So the MIG and TUP system, they are what are controlling transcription based on glucose levels. Okay? So the repressor is on galactose levels. These guys are on glucose levels.
So they are negative control.
But these are negative controls. Yeah.
So when the top one and the MIG one dimerize, what they're repressing is the synthesis of galactose. Is that what that
Of genes that are going to be made into enzymes. Right. So the mRNAs, the genes will be made into mRNAs, which will be made into enzymes that can synthesize, that can break down galactose. Exactly. Because galactose is present. Right? Yeah. No, he just sits there. Okay. So in this case, galactose is present. Yeah. Oh, so in this case, glucose is not present. Okay. So he's not going in. Now we need to look at our galactose levels, and we're like, oh, if galactose is present, then we have our 4P, our Gal4P, and our Gal3P activating this guy with the Gal80P falling off. Okay? So one's looking at galactose, one's looking at glucose. Yeah.
So is glucose repressing MIG-1
It's
from being phosphorylated?
from being phosphorylated. It stops it from being phosphorylated.
And then MIG-1, just like you said, represses top one from continuous transcription.
TUP1 is a repressor.
Oh, top one, sorry.
Yeah, so TUP1 is a repressor, but he's not bound. He can only bind. MIG1 is a repressor too. They have to dimerize together. This goes back to like the combinatorial heterodimerization. Like they dimerize together. They dimerize together, and they are the repressor together. Both of them bind to the DNA and turn off the gene. Okay, and this happens because MIG1 is not phosphorylated because glucose is high. Okay, but if glucose is low, it's phosphorylated. He can't enter. He can't dimerize.
So this step precedes the other step that we just talked about.
Okay, these steps happen simultaneously. Yeah. So you can have the repressor fall off. You can have the repressor bind, and you can have an activator bound too, because one is controlled by glucose and one is controlled by galactose. Repressor is controlled by glucose. Galactose controls the activator.
So the repressor's bound, and I want to read these things.
That's correct. Yeah.
Low glucose transcription will happen.
If galactose is present. Yeah.
What is the control? So big one and top one controls the glucose transcription.
MIG1 and TUP1 sense if glucose is present.
But what are they controlling? What are they?
They are repressing this gene, these genes that make enzymes that break down galactose. Okay, so we only want to make those genes if glucose is not present and if galactose is high. So MIG1 and TUP1, if glucose is present, there's no reason to make those genes. So they bind to the DNA together, and that turns off transcription. They are a repressor. Okay? But if glucose is not present, then MIG1 gets phosphorylated. He can't enter the nucleus. And if he can't enter the nucleus, they can't repress. Now we need to see, is galactose present? If galactose is present, then the activator allows transcription to occur.
So if the lactose is present, then it's how is it, GAL?
The Gal4P.
Gal4P and Gal3P will just synthesize.
Exactly. But if galactose is not present, then Gal80P and Gal4P are bound together. Okay. That's the Galoperon. That's the only one you need to know for the eukaryotes. Okay, so eukaryotes are, of course, more complicated. And so here's some little details. They have a nucleus. So the mRNA, it has to be exported from the nucleus, and it has to be able to survive in the cytoplasm. And that's the reason that we do modifications to the 5' end of the mRNA and the 3' end of the RNA. So we put a cap on it and a tail onto the RNAs. And these modifications happen at the C-terminal domain of the RNA polymerase II. So the C-terminal domain is like a table that you can prepare everything out for. Okay, so here's the capping and the adding of the tail. So capping, this is just putting on the 5' cap, which is an upside down 7-methylguanosine. Okay, so it's facing the wrong way. That is the cap, and the cap helps with translation. Okay, so it's going to help. So here's my DNA. We've made our mRNA. This guy has a cap. He has a tail. The cap is going to help in translation. So the ribosome, taking the mRNA and making a protein from it. Okay. The cap is added using the cap binding complex. Okay, or CBC, which is right. I'm sorry. I'm skipping a step. So after the cap is added. So the cap is going to be added with the capping enzyme. Then we have the cap binding complex, which keeps the 5' end attached to the C-terminal domain. Okay? So then we get more synthesis of the mRNA. And then we're going to add our poly A tail at the 3' end. Okay? So this happens in this order right here. We have these polyadenylation factors that are going to bind to a site before transcription is completed. Okay? So you have my RNA polymerase. He's continuing on. Okay? He has this C-terminal domain here. Okay. So before this guy even stops, he's going to continue on, but our polyadenylation factors are going to bind before he finishes. Okay? That's pretty much all you need to know about him. And he signals for the endonuclease to come and cut. So an endonuclease comes and cuts this growing strand of mRNA. Okay, so even though we're still growing over here, it cuts it right here. And then we have PAP, that stands for poly-A polymerase. And that's going to just insert the poly-A tail onto this growing RNA strand. So here's my 3' end, here's my 5' end, here's my CBC. Okay? Do you all see how that's happening? Yeah.
With the capping enzyme, then go and put a cap on that new end part?
Right here?
You just cut the mRNA.
I cut the mRNA on the 3' end.
Yeah, so the 5' of that, now that's still in the polymerase with the 5' capping.
No. So this piece right here, it's not going to do anything. They'll just, yeah. So we cut it out, and then the PAP, the PAP, poly-A polymerase comes in, and it adds a polymerase. It adds a poly-A tail, and it does it without a template. The only thing it knows how to add is A's, templateless. Pretty cool. And so the poly-A tail, it helps in exporting this mRNA from the nucleus. It adds stability, and it helps it bind to the ribosome. Okay, so it helps in those three things. Okay, questions? Yeah.
There's a lot of details in this. Like, how in detail should you know this process?
This. Yeah. These are things that I wrote down. Like if I took the time to write it out, then yeah, you need to know it. Yeah. Okay. Yeah.
Maybe this isn't important, but PABP is the same thing that you said, like PAB adds the tail and then there's one that extends it. You know that?
Maybe ask him a little more about that. But PABP stands for poly A binding protein. And what happens is now that you have the poly A tail, you get PABP that binds to the poly A tail. And it just adds to the stability so that it doesn't get degraded. And the reason why is because out here in the cytoplasm, we have these little enzymes called RNases. Okay? And our RNases, they eat up mRNAs, right? Because we don't want the mRNA to last forever or we would get endless amounts of proteins. So the RNases are going to eat them up. So they work on the tails, trying to eat them up. So PABP just protects the tail. Okay? So all of these guys right here, the endonuclease, polyadenylation factors, I mean, this is all happening at the C-terminal domain. Okay. So for splicing, so our RNAs that are being made in eukaryotes, they have these introns, okay? So there's junk DNA. So most of our DNA is filled with introns that get put into the growing mRNA. And so we need a process to cut the introns out. And so we use these SNRPs, so the SNRMPs, which are actually RNAs that help cut out introns, okay? So the SNRPs we talked about are U1, U2, U4, U5, and U6. Yeah. The exons are the things that have the genes on them. They're the ones that are exiting the nucleus. Okay, so here's the steps. You won't have to draw this out on the test, but you need to know what each of the U's do. So here we have U1 and U2, those are binding. One of them binds to the branch point, that's U2, right? And you have U1 that binds to the exon 1 at the 5' end, or the 3' end of the exon, but the 5' of the intron. Yeah, so those bind. So these are RNAs. See how the RNA is binding there? So U2 binds to branch point A. And then we have U1 being replaced by U5 and U6, okay? And when it's replaced, U2 is going to guide branch point A to attack right here at exon 1, okay? So now we are forming a bond between branch point A and this G right here, okay? Do you see that? So then the question becomes, what is this OH group? Because we have the 3' connected right here. We have the 5' OH group connected right here. What OH on this branch point A is bound to this G? This is the 2' OH group, right? Because RNA has a 2' OH. So it has 3 things that are bound there. So now we've catalyzed this attack. So now exon 1 is kind of willy-nilly, but it's being held by U5. So now we're going to have this free OH group on exon 1. This is the 3' OH group, right? Because branch point A has cut right here. Now we have an exposed 3'. The 3' is then going to attack exon 2. So that joins together exon 1 and exon 2. So that's what U5 is there doing. Yeah, and then it gets ligated together, and the lariat, which is now no longer connected because this attack's right there, it floats away. So the intron is the lariat that floats away. Okay? Yeah.
So just get to know the boxes again for this slide.
Yeah. Grab a neighbor, walk them through it, but you don't have to reproduce it. But you will see multiple choice questions on it. Yeah.
Okay, you said towards the end there, the 3' OH group of exon 1 attacks at the 3' splice site, which is the 5' of exon 2.
Yes.
Is that right?
Yes. So this 3' right here, this is the 5'. It's the 5' location. So remember, it has the phosphate there. And the 3' attacks the phosphate. So the oxygen is acting as the nucleophile. Yeah. Yes.
This might not be something that you know or that even really matters, but why does it use a branch point instead of just pulling from either side, like either splice side and pulling together and cutting there?
You mean?
Because isn't the branch point in between the two splice sides? And so the branch point will move to one splice side and then move to the other splice side?
Right. So you do it in this way so that you don't need to really know, but this is the way that you can have exon 1 bind to exon 2, because if you try and have this guy connect here, the primes will be a little off, and so it won't actually bind correctly. Okay, so you won't get the correct binding. So the branch point A is important because that has a 3' hydroxyl group. Not a 3', a 2' hydroxyl group. Okay? And that can do attacking. Yeah. I don't know what else to really say. Yeah. But if you go through and you work out, look at each of the primes, if you try it your other way, it won't work out. Yeah. Other questions?
How do you get 2 primes?
You mean on the activated A? Yeah, so RNA has a 2' hydroxyl group. It's just there already. Yeah. Yeah, so it's just floating there. It's not connected to anything. So it's free, and so now it can attack at the exon 1. Okay, between exon 1 and the intron. Okay. Okay, so RNA types, we have mRNAs. They're the ones that carry the genetic information to a ribosome. We have tRNAs. They carry amino acids to a ribosome. So you're not going to have to draw this, but you just need to know the basic definitions. But here's my DNA. Okay, we've made mRNA from my DNA. The mRNA comes over here to the ribosome. Okay, let's have him going the other way. Okay, so he's going to come in through the ribosome. The ribosome is going to have these 3 positions, and we'll learn about them next time. But what happens is we have these tRNAs. We have these tRNAs that are connected to amino acids, and the tRNAs, they have nucleotides because they're made up of RNA, right? And these nucleotides will match up with the mRNA, okay? So we get these tRNAs with these amino acids on top of them, okay? And they start to bind together, and these guys move in this direction, and so you start to make this protein of amino acids. So the tRNAs bring in the amino acids. mRNA is what's carrying the genetic code to the ribosome. You have ribozymes, which are RNA molecules that act like enzymes. So they're not enzymes, they act like them. They have enzymatic properties. So examples of that are rRNA, which makes up the ribosome itself. You also have SNO RNAs. Those help direct modifications to the rRNA. Because, once again, here's my cell. Here's my DNA. We use a different RNA polymerase, but transcription of rRNA is going to be the exact same. And so once we make this rRNA, then we need to modify it so that it can form the ribosome. Okay? So the catalyst that does that is SNO RNase, which is also an RNA that was made in the nucleus by an RNA polymerase also. Okay? And then we have our snRNAs, our SNRPs. They're the same thing, which we talked about up here, which was part of splicing. Okay. Combination of transcription factors dictate the body plan. Yeah. So I think this is our last slide. No. Almost. So I'm going to be real quick with this and give you the very basics. So if we have a snowman, okay, yeah. Okay, so our snowman, there's these genes, okay? So our snowman, he's made up of cells, okay? Obviously. So he's made up of all these cells. So what happens is you have a snowman mama and a snowman dada, right? And you got this yeah, except for it's a snowman one. And he finds the egg, right? Okay. So he finds the egg, and we have the same DNA in our egg, correct? Okay. Now this egg is going to start to divide, and then we end up getting a bunch of little cells, okay? And these cells start to differentiate. Like if you take embryo, whatever it is, think about embryos. It's going to divide. You're going to get the 3 tissues that start to form. Then you start to form more tissues from some of those tissues, and you get this differentiation that's occurring, okay? The DNA in all of those cells is the same though, okay? And so the idea is that this DNA is going to be expressed differently in each of those cells. The mRNAs that are being made are going to be expressed differently. So we have this gene, it's called even skip, okay? And even skip is a gene found in all of these cells that's going to dictate what body part forms in a fruit fly, okay? So depending on how even skip is expressed, how much it's expressed will determine what happens to the fruit fly, okay? So if even skip right here maybe is expressed a ton in these cells will become a head. If it's expressed a ton in these cells, those cells will become feet, okay? Sometimes we have mistakes, and maybe it's expressed just a little right here, but a ton right here, and we get a leg coming out of the eye or something like that, okay? So that's even skip. It's a gene that controls the development of the body. Now, even skip, its expression is controlled by transcription factors, okay? And in class we talked about bicoid, crouple, hunchback, and there's one more, giant, yeah, okay? So we have 2 of them being repressors, 2 of them being activators, okay? And the combination of them, their concentrations in the cells, will control how much even skip is expressed. This is combinatorial control right here, okay? So what is the big takeaway from this? The big takeaway is that you probably don't need to, so the big takeaway is you don't need to know these names. You don't need to memorize which ones are activators, which ones are repressors, but you need to understand the concept. So I'll give you a little example. Oh, sorry, here's my snowman. Okay, so we might have a similar gene that controls how the body parts are made. Okay, let's say carrot. Okay, so carrot is right there. Okay? So there's a gene called the carrot gene. Okay? And the carrot gene, when it's expressed, you're going to get a carrot forming. Okay? And so let's say that we have some repressors and activators. So in this case, we'll have, we'll have like the coal activator. We'll have the stick activator. And we'll have the marshmallow repressor. Okay? So the idea is that where you have high concentrations of the activator and low concentrations of the repressor, you're going to have the carrot gene expressed. Okay? So this snowman I've drawn next to this graph here, and right here I'm going to put the concentration. Okay? So my concentration, what am I doing? There we go, is increasing. Okay? And this is the distance. So we had cells. Remember, this isn't the snowman yet. He's just a group of cells that need to be differentiating. Yeah. Of the activators and repressors, to see how much of the carrot gene is going to be expressed in different regions. Okay? So maybe, in this case, we have a carrot. We know it's going to go right there, right? So we would expect to see maybe something like this. This could be like my coal activator. Maybe we have an activator like this. This one is my stick activator. And then we have something like this, which is my marshmallow repressor. Okay, so you see that where the activators are high and the repressor is low, we get a carrot. Yeah? Cool, right? So then let's say that we did this. Okay, so let's change this up a little. Let's say that here's my coal activator. Okay. Here is my carrot activator. Yeah. I'm sorry, it's stick. Thank you. So here's my stick activator. And let's say that I have a repressor here. Okay, and this one is my marshmallow, right? Okay, so the question now becomes, where up here would you get a carrot? Yeah, so you'd get a carrot right here where both activators are high, but we also have another place where an activator is high and the repressor is low. You might not have both of them, so you might get like a little bit of a carrot there too. Okay? So like that's the whole deal with like how transcription factors, they can affect these genes that lay out the body plan of something. Yeah.
So is this more of the multiple choice question?
You won't have to give your own example, but this probably wouldn't be a multiple choice question. Yeah. Okay. Any other questions? Oh, we're not done. We have one more thing, epigenetics. Okay, here's the basics in epigenetics. We can do modifications to the DNA's histones or to the DNA itself that can make it be expressed more or expressed less. And these modifications can be passed on to our children. And so one indication of epigenetics is that there's no mutations to the DNA. The DNA is not mutated. So any changes you see between the parent and the child, you won't see any mutations there. There'll be none. Okay? You're just modifying the DNA or its histones. Okay? Acetylation of the histone tails causes more... Yeah?
What are you talking about modifications to the DNA?
modifications as in like methylations or acetylations or simulations or just like all of the giant list of modifications, but you're not actually changing the base pairing. Yeah. So if you acetylate histone tails, that's going to cause transcription. If you methylate it, that's going to cause repression. And then also, I'm not 100% sure because I don't know if it was in the slides or not. I was looking through them. But I think in class tomorrow you should ask him about how you pass on the epigenetics to the children. Like, what are the means by doing it? Like, I can describe one of them to you, but I'm not sure about all of them. So ask him about that in class. Okay? Yeah. Okay, so that's everything. Any other questions? No? Okay. Great. Go home, go to bed. Really study the PowerPoint, okay? PowerPoint has pretty much, I mean, if you look at my exam, so this is the exam I took. So, like, I have my little, like, checks next to each question. I've never seen so many people, like, squint before. But, yeah, I have, like, my little checks. That's because as I went through and added something, I checked off the question that I had answered with my PowerPoint. Okay? So, but like I said, there's that one question that I said you should ask Dr. Poole, like, really ask him that because I don't know exactly. Yeah.